Good afternoon. Thanks for being here today. I don't know, my expectations for the attendance of this seminar was not too high, so I'm very happy to see you, all of you here. And we have a lunch seminar. My name is Sira Echevarria. I'm going to be your chair today. And I have with me Cathy Marulli from ETH. She's a member of Paola Picotti's group. And we have Erwin Schoof. Erwin Schoof. He is an associate professor at DTU, and he's very well known for his research in single cell. And this is what you can expect today. So I will give a brief introduction, and then Cathy will talk about a workflow that they develop in her lab. It's called FiLip, and it's for studying dynamic protein complexes. And Erwin later will talk about the advances in technology, hardware and software that are pushing the potential of single-cell proteomic research, and of course how he is using them. So at Biognosys, we aim at providing proteomics solutions for anybody that wants to do proteomics, and that is independent of whether you guys have access to a mass spec facility or not. So we have two different type of solutions. On one hand, we develop software and kits. That's probably in this audience why we are mainly known. But if you don't have access to a mass spec facility, or your facility lack the expertise on particular applications, or simply there's no capacity to run certain projects, we also offer CRO services. We can do those things for you. And this, what you see here, is our whole portfolio of software and kits. I want to highlight PQ500. That's peptide kits, heavy label peptide kit for targeted proteomics on plasma. And we also have the IRT kit, which is used for your QC endeavors in a mass spec facility. Then we develop software for mass spectrometry data analysis, and here I want to highlight Quick. Quick is our software for monitoring the performance of your mass spec facility, and the nice thing about it is that it's vendor agnostic, so even if you have a hybrid facility with instruments from different vendors, you can use Quick. It's a very nice to use tool to monitor the whole facility. And Quick is for free with the condition that you use the IRT kit in your samples. Our technology, what we offer, our solutions are based in three main pillars. One is performance and scalability, so we strive at providing the best performing tools for the applications that, for any proteomics application. Reliability and robustness of the results you get is non-negotiable for us, and we put a lot of efforts in doing very intense benchmarks before we release anything to our customers. And user experience is also a very important aspect for us, and we put quite some effort on that as well. So Spectronaut is our solution, the one that's going to be featured in this seminar, is our solution for DIA proteomics, probably the one you know. If you know any product from Biognosys, is probably this one. And to highlight, we have probably the best performing library-free solution out there. But we also have a solution for targeted proteomics, so that's SpectroDive. And one thing to highlight about SpectroDive is that it is a very good solution for large sample cohorts, large panels, and you get scalability and high throughput in targeted proteomic experiments. Then we have SpectroMine. SpectroMine is the solution you would use for anything that you acquire in DDA mode, and this is TMT or label frequentification in DDA mode, or if you just want to search and generate spectral libraries, for example. SpectroMine is very fast, and you also get more IDs than the main competitors out there. So if you want to try them for yourself, we provide free access to them for trialing purposes. We also have preferential pricing for academic institutions. And if you get more than one of our solutions, you also get a very nice discount. So if you're interested in trying them, please reach out to us at software@biognosys.com. And with that, I want to invite our first speaker to the stage, Cathy.
Hello. No. Thank you so much. Yeah, thank you very much for giving me the opportunity to present my project here. It's about probing interactome dynamics, and we do it with FiLiP-MS. So proteins, they often interact with each other to form protein complexes, and we all know that they perform a number of essential cellular functions, such as translation, signaling, or cell cycle control. And there are a number of tools with which we can study those protein complexes, such as affinity purification, mass spectrometry, yeast-2 hybrid screens, and coelution-based profiling. Now, from these powerful methods, we learned that proteins form these densely interconnected networks that can integrate information about the environment and genetics and lead to a very specific cellular response. And the question that we try to address in this project is, can we monitor the dynamics that are going on in these networks? As I told you, we have these methods, but all of those are very good when it comes to mapping protein-protein interactions, but they become extremely tedious when it comes to probing the dynamics, because, for example, in ETH, you would need to have one pull-down for each protein that you're interested in. So we were thinking, is there a method with which we can actually do this in a scalable way? And there is a method that was developed by our lab, which is called limited proteolysis coupled to mass spectrometry. And this method, in general, identifies structural changes, one of them being the formation of a protein complex. So imagine that in one condition you have a monomer, and in the other condition you have a protein complex. We now apply a limited proteolysis step, and the protease can cleave surface-accessible residues. Now, in the monomeric form, the cleavage can occur in the protein-binding interface, but in the complex-bound form, it cannot occur because their sterical hindrance of the interaction partner does not allow this. And these signals we can measure by mass spectrometry. Now, the problem with this is that this is not the only signals we measure, but we can also capture binding of nucleic acids, small molecules, or PTMs. So how can we do this? We need to find a way with which we can specifically fish out the fingerprints that come from protein-protein binding events. And this is exactly what we address with our novel approach called FiLiP-MS, which is called filtration coupled to LIP-MS. So the idea is quite simple. You take a yeast lysate and you separate it by size through a serial ultrafiltration process with molecular weight cutoffs ranging from 10 to 100 kilodalton. And this then results in large protein complexes being present in the high molecular weight fractions and the monomers in the low molecular weight fractions. We then perform LIP-MS on all of those fractions, and again, in red, the protein binding interface will be cleaved in the monomeric form, but not in the complex bound form. We acquire all our data in DIA and do all the analysis in Spectronaut, and by doing so, we identify 6,441 markers coming from 1,068 proteins. Now, first of all, we need to confirm that this library is indeed enriched in protein-binding interfaces, and we did this by 3 ways. First of all, we did a domain-based interpro-enrichment analysis, and what is very nice here is that the most abundant significantly enriched term is indeed protein binding, directly followed by RNA or nucleic acid binding. And this makes a lot of sense because our method is not able to distinguish between a protein-protein complex and a protein-RNA complex, which are also fairly abundant in the cell. Next, we mapped our markers onto PDB structures. So here's an example. Here I have a trimeric PDB structure, and in yellow you can see the interface. If we now map our hits onto this protein, then you can see that in blue, the markers are overlapping with the interface, and other detected peptides in black that they don't change between different filter fractions, so between monomer and complex, they are located further away. We performed this analysis globally on all available multimeric PDB structures and did a ROC curve analysis, and we found, depending on how we choose the parameters, an AUC between 0.61 and 0.71. This tells us that our markers are better than guessing at random, where protein binding interfaces are located in PDB structures, but it is far from perfect. And there are many reasons for that. One of them is, of course, noise in the data, but then also we capture protein RNA binding interfaces which are not part of this analysis. And the real question that we have to ask is the PDB database really the perfect ground truth? Because every novel discovery that we would make and is not part of this database will be rated as a false positive in this analysis. So what would be the optimal experiment? Well, it would be to mutate one of those binding interfaces that we found and see that the protein cannot assemble anymore. This would be a lot of work, but luckily there's a database out there with manually curated sites. If you mutate them, then the protein complex cannot assemble. And what is really cool is if we look into this database, in 82% of the cases, we have a direct overlap between our marker peptide and the mutation site that is known to disrupt protein-protein complex formation. All right, so I hope I could convince you that we have a library that is enriched, at least in protein binding interfaces. How can we now use this library to track dynamics of protein complex formation? We combine this now with standard FiLIP-MS experiments where we get these complex structural fingerprints for all kinds of structural changes. And we chose to do this under hydroxyurea stress because under hydroxyurea stress, yeast form P-bodies, which are the small white dots that you can see. And we know that a couple of protein complexes need to form in order to make those P-bodies. And one of those protein complexes is actually the interaction between PAT1 and DHH1. So again, we performed FiLIP-MS on the control and hydroxyurea condition. We get these complex structural fingerprints. We did all the data analysis in DIA and the Spectronaut. And if we now look at the results for this PAT1 molecule, there's no structure available. So this is now the sequence of the protein. The only thing we know is that in the N-terminal, we have the location of the PAT1 DHH1 binding site. Now we can look at our peptides. We have a couple of non-changing peptides all over the sequence, and then we have our LIP peptides. These are the peptides here in blue. And these can now come from all kinds of structural events that are shown over there. But the cool thing is now that we can make use about the information of the library and check which one of those LIP changes corresponds to a protein binding interface marker change. And if we do so, it is very cool that the only LIP peptide that is part of our marker library is directly located at this PAT1 DHH1 binding interface. And this is by far not the only example that we have for such protein complexes. We have another number of protein complexes, such as the LSM17-PAT1 complex or the XN1 complex, where we also see significant protein binding interface marker changes that are directly located at the binding interface. All right, so I hope I could show you that we can actually capture known protein binding interface rearrangements with this method. But now you would end up with a very long list of proteins that change their binding partners. But our method cannot provide you any information about who is actually binding in the partner that is changing. So this is why we choose to integrate our data with prior knowledge. So we take protein-protein interaction networks. And the idea is that if you have multiple subunits of one cluster of one protein complex changing at the same time, then it's likely that this complex is undergoing a change. So how does that look like for the real data? We take a protein-protein interaction network. In our case, it's the complex portal database. Then we map our markers on there, the changing markers. We propagate the signal by the PageRank algorithm. We extract the part of the network that is closely connected to our hits. Then we cluster it, and we can nicely rearrange it and see which protein complexes are actually changing. So now here, this is more than 50 protein complexes. Some have one changing subunit, some have up to 10 changing subunits. Of course, the more subunits change, the more confident it can be that something is going on. And this is actually the end of the pipeline, but this is also the most cool part because now you can dig a little bit deeper into biology and see what is going on, and this is exactly what we did at this point. So one thing that struck our attention is this SAGA complex. SAGA is a very large and complicated complex with many functions, but one of them is that it has a histone acetyltransferase activity. This is this GCN5 molecule up here, and it is known to transfer it's known to acetylate histones. Yeah. So first of all, we wanted to check whether this is active under the stress. So we checked for acetylation changes in our data set, and we could confirm that we have increase of acetylation in histone HTB1 and HTB2, so telling us that this GCN5 acetyltransferase is active under this condition. But why am I telling you this? Well, it does not only acetylate histones, but it's also known to acetylate a number of other proteins. And actually, out of those 56 complexes that we identified, 12 of them have targets of this GCN5 acetyltransferase. So the question we were asking is, are those protein complex rearrangements dependent on this GCN5 activity? How could we test this? Well, we created a mutant with a single point mutation in the GCN5, and this could then no longer acetylate in general. And what you can see is on just doing a spot assay, the mutant is really struggling to grow under this hydroxyurea stress. And now what we can do, because our method is quite scalable, we just repeated the whole workflow again with the mutant. So we grew the mutant under control and stress. We checked which protein binding interface markers are changing. We did network propagation and extracted the protein clusters that change. And now we can compare the protein clusters that change between the mutant and the wild type. So if we look into this data, first of all, we have quite a substantial number of protein complexes that change the same way in the mutant and in the wild type, suggesting that they are not dependent on GCN5. And also telling us that our method is kind of robust to identify a subset of complexes that change similarly in two different data sets. Then we have a couple of protein complexes that change more in the mutant and in the wild type, but the most interesting part is that there's a very large number of protein complexes where we see more changes in the wild type than in the mutant, suggesting that those are dependent on GCN5 activity. And what makes this even better is that if we now look where our GCN5 targets located, these are the green boxes here, and you can see that 10 out of the 12 protein complexes that rearrange more in the wild type are known GCN5 targets. Now, we wanted to go a little bit deeper, and something that struck our attention is this LSM17PET1 complex. I already talked about this before. It is a complex needed for P-body formation. So we were wondering, well, does GCN5 activity somehow influence the formation of P-bodies? So we just did a very simple microscopy analysis, and we stressed mutant cells with hydroxyurea. And what you can actually see here is that the mutant cells, even without stress, already have tiny P-bodies present. If we then apply the stress, we have larger or more P-bodies, and if we take the stress away again, then the mutant actually needs longer to recover. All right, so can we now track protein-protein interaction dynamics? Well, I proposed you the following approach. We first created this library of peptides that are located at binding interfaces. I'm not going to lie to you, it takes quite some work to make this library, but the good thing is you do it once per organism and you can use it over and over again. And the library in itself is a very nice standalone resource because it has potential novel binding interfaces that can be useful for modeling or drug design. Then I showed you how you can use this library in any kind of FiLiP-MS experiment that you're interested in order to fish out which protein binding interfaces change. And this makes the whole approach scalable. You can screen multiple conditions on a proteome-wide scale in a couple of days. So for example, in the lab, you just need 2 days, and then you need roughly 1 day of mass spec time for the acquisition. And last but not least, I showed you how you can integrate this data with prior knowledge in order to generate hypotheses on the protein complex level. And in our case, we could link GCN5 activity to protein complex rearrangements and also GCN5 activity to P-body formation. And with this, I would really like to thank Biognosis to give me the opportunity to present this work here. I want to highlight Chris, with who I've been working on this project for the last 2.5 years, our 2 collaborators, Tom and Norbert, my professor Paula Picotti, and my 2 research group that I'm part of, the Picotti and the Leimer group. And thank you for your attention.
Thank you, Cathy. And now I want to give the stage to Erwin Schoof. Thank you.
Thank you. Well, thanks for that introduction. Very kind, and the invite to give me another opportunity to present some more of our single-cell work. I promise you there's not much overlap with the previous session if you were here. Basically, indeed, we really are seeing this clear shift towards DIA-based approaches to single-cell. We welcome all the kind of computational tools that are being developed, including Spectronaut, of course. So I hope to show you some of the ways that using Spectronaut has helped us improve our workflows. So I mean, some of you might still not be convinced why you want single-cell technologies, and you might have heard the smoothie comparison or pizza, and actually I also like this one. We have a big forest around our house, so we see a lot of these ant heaps. And what if you were tasked, I mean, it looks very simple, but inside it's actually rather complex. You have lots of different larvae, a queen ant, different kind of worker ants. So what if you were tasked with the challenge to study the proteome of the queen ant? Would you take an approach of putting this ant heap in a blender, running proteomics on that? Or would you rather take tweezers and take the long and arduous task of taking this apart piece by piece and running proteomics on that? This kind of just to draw the analogy to the blender part being traditional molecular biology. We used to do this with tumors and primary tissue a lot, whereas, of course, the tweezer approach is what we can do with single-cell work. Of course, we're not as interested in studying ant proteomes, but I really like the analogy of seeing this as a tumor, and we want to find the cancer stem cells, which is a very low population in these tumors, and this is really why we need single-cell technologies. So as I also said before, I mean, we really want to better treat patients and understand the responses of individual cell types in these tumors, and we know for a long time now that these tumors are actually very complex cell populations with many different cell types. And chemotherapy is very good at targeting cancer cells because they rapidly divide, but there's many other cell types that do not respond as well because they might be quiescent or not as actively dividing. And to really capture this intra-patient heterogeneity, we want to start the initial steps towards the next state-of-the-art in personalized medicine. So not only taking into account the individual's makeup, but actually also every individual's tumor's makeup of cells. And this is really one of the objectives my lab is trying to chase using single-cell omics methods, including proteomics. And that's why we need to really keep developing a lot of single-cell technologies, because proteomics is lagging behind, but it is really taking off with more and more speed, which is very exciting to see. I often also get asked, and probably not as much in this audience, but why bother doing proteomics? We can run tens of thousands of cells with single-cell RNA-seq. It works well, it's relatively cheap, deep coverage. But and I'm sure many of you agree that this is only a black and white picture, whereas at protein level we can really add color to this, and this, for example, blood hierarchy that we heard earlier from Jenny Henson as well. And we really want to study how do cells decide which branches to go down. Now, we used to think it was a kind of discrete cell state, different cell populations, but it truly is a continuous or developmental continuum, meaning we need single-cell methods to learn how these cells are. Now, we all know that the correlation between mRNA and protein is rather poor in mammalian systems. And this, of course, due to protein translation, degradation rates, and something that mass spec will be the only technology to truly capture our PTMs like phospho and ubiquitination, etc. So this is why we're really developing a lot of technology to do single-cell proteomics by mass spectrometry, or SCP-MS. Because in principle, flow cytometry microscopy is already doing single-cell proteomics, but we want to make sure people realize this is by mass spec. So there's been really a ton of work, and probably this slide is already a little outdated again, because they seem to be coming out every week. But there's really been two main camps so far, TMT multiplexed and label-free. As of recent, we now have very exciting developments in multiplex DIA. And if you ask me, this is probably where the future is at. Short gradients, high multiplexing with DIA. And I just put 3 kind of key papers in this space if you want to read up more. Of course, today I will basically focus only on DIA with Spectronaut on label-free. And I just wanted to showcase 2 approaches that we explored in this space. The first one's published on linear ion trap-based data-independent acquisition. The second part is based on a new Astral instrument. So the linear ion trap, for those of you who are less familiar, this is, by the way, a really good source if you want to know a lot more. But basically, I just compared the Orbi versus the linear ion trap in a few features. Like the Orbi is very high resolution, the LIT is very medium or low resolution. You have very high mass accuracy in the Orbi trap. You have not as high mass accuracy in the LIT. Conversely, you have quite a bit higher sensitivity in the LIT, about 10x compared to Orbi. whereas the Orbi is medium-high sensitivity, and it's slower in the Orbi, whereas the lit is much faster. And this is why we really thought this high sensitivity part could help us a lot with single-cell proteomics. And just to point out here, I mean, when we started these experiments, Diane was just gaining traction, so we thought, okay, we can just run free software. But actually it didn't really produce any hits, but we just got a copy of Spectronaut and put the data in and immediately got beautiful results. So just to stress that Spectronaut really seems to be the most versatile search engine for DIA data based on alternative mass analyzers. So we used the linear ion trap as built in the Eclipse tribrid instrument, but also the Fusion or Lumos or now the Ascent, where we combine the Orbi with the linear ion trap. So we do all MS1s in the Orbi to keep that high mass accuracy, but to gain the sensitivity from the linear ion trap here. So to kind of get an initial survey going, we basically decided to scan 500 to 900 MZ with 10 MZ windows, so 40 windows in total, and comparing the linear ion trap at different scan modes and the Orbi trap at different resolutions with corresponding free ion injection times based on the transient of the Orbi trap, and kind of matching the injection times based on, can't really call it transient on the linear ion trap, but the similar scan rate, so the free injection time for these ions, with very similar cycle times between Orbi and linear ion trap to really compare how are these two mass analyzers performing at 10m/z windows. So of course, you heard me before, those of you who did, and we're really into wide windows now, but that doesn't really work on the linear ion trap due to the lower resolution. So it's really to compare apples and apples on two different mass analyzers. So our first assay was to run kind of high load down to low load. You can see at high load, the Orbi traps methods are clearly outperforming the linear ion trap, but as soon as we dip below 10 nanogram, you can see that the Orbi trap sensitivity starts to take a hit, and at that point, your linear ion trap starts performing better. Kind of summarizing that in a different way, I won't go in through all these, but just to kind of take away the picture that on the top, with the linear ion trap, we can see that for all loads, we're getting quite a few peptide IDs, whereas for the Orbi trap, we're only seeing these big bars on the high loads, and at the low loads, we're really not gaining many IDs, suggesting that the lit is a good analyzer to pair for low input analysis. If we look at the types of peptides we're identifying, and then saying in gray these are identified only in the lit, and in red, lit and Orbi, you can again see that only in the high loads of 100 nanogram, Orbi is performing well, that as we go down in loads, we see the lit kind of taking over performance-wise. You might ask, well, what about the quant, and in this case, precision? So here we're comparing Orbi trap abundance versus lit abundance, so comparing both mass analyzers for these runs, and you can see that they're actually very much in agreement, except maybe in these 1 nanogram, where we have the Orbi trap running at the lowest resolution, shortest injection time, and it's very clear that the sensitivity is insufficient to identify and quantify enough peptides, but that, at the other ranges, it's actually looking very good in terms of lit quant quality compared to Orbi trap. Just looking at this in a different way, comparing two replicates, either for lit or OT, you can see that both mass analyzers are very reproducible if you inject the same sample twice and just do a simple correlation there, suggesting that the quant is also accurate. So conclusion was really here that lit-dia is a capable tool for low input proteomics if you want to keep isolation windows narrow. In the case of post-translational modifications, you might not want to go wide window because then you get a lot of B or Y ion overlap between modified and non-modified. So this is where this could be a use case for it. We just published using also the lit as the MS1 analyzer if you want to read more, but I won't go into detail today. So then we thought, let's also try and apply this. And we are getting excited about this concept of laser capture microdissection towards single cell. We're not there yet. But we basically cut out different areas and then kind of looking at the number of peptides we identify in our cuts, clinical samples, compared to HeLa. You can see that HeLa is much easier to identify a lot of peptides, whereas you need a lot more kind of equivalent input material from clinical FFPE slides to get similar proteome coverage. So just to clarify why trusting HeLa data is not always relevant for your biological experiments. So we did this titration series of different surface areas corresponding to these kind of cell numbers. You can see that from about 130 cells onwards, we're approaching these 2,000 proteins that we can measure, and we start kind of leveling off for the lit style experiment around 130 to 260 cells. That seems to be a decent approach for trying to do this low input spatial proteomics. And we're basically picking up a variety of lymphoma biomarkers across the intensity range. So then to the last part, how about another mass analyzer that was very recently introduced at ASMS, the Astral. Won't go into too much detail, but just to clarify, this instrument also has an Orbitrap, so the Orbitrap Astral MS, and then the Astral mass analyzer at the back end, where ions can fly through and then get detected at the end with very high sensitivity. So here we first had to figure out how to actually run DIA on this instrument, because in terms of sensitivity, it's reminiscent of the LIT, but in terms of resolution, it's much higher. It's about 80,000 resolution on an Orbitrap scale. So we get high resolution, high sensitivity, both in one. So first we checked with 1 nanogram and 250 picogram how much injection time we need. And we can see 20 to 40 milliseconds seems to reach a sweet spot, which is, like I said before, a tenth of what we normally need on Orbitrap. Looks similar to what we were using on LIT, but now we get more resolution. So if we compare kind of OT DIA on Eclipse and Astral DIA, we can see that we are reaching more hits in this low abundance while keeping CVs low. So we are detecting lower abundance proteins. From 1 nanogram, it's kind of interesting to see that just in the less than 10% precision on Astral, we're capturing already more proteins than in the entire data set on Eclipse, running the same gradient length, same column. So it really suggests that this instrument is much more sensitive. And that also allows us to dig deeper into the low abundance proteomes. So here we're just comparing Eclipse and Astral again in green and red. And if we overlay kinases, you can see we pick up a lot more in the Astral. We pick up a lot more transcription factors, which are key cellular decision-making proteins. And to be able to do this out of 1 nanogram and less got us very excited. There are some preprints about the technology. So can we do single cell on Astral using our WISH DIA protocol, using Spectronaut for searching, that we basically deposit single cells in 3D 4-well plate, run them label-free one at a time, using the Vanquish Neo, the micropack low-load column that was recently released, and to just run these on 18-minute methods from injection to injection, so about a 9-minute gradient, active gradient. And this was all done with Spectronaut 17, direct DIA+, so no library used to boost the numbers. This just to show the peak width of this column at these short runs, so about 3-second full width half maximum, so very sharp peaks, and we get good quantitative accuracy with at least 6 points per peak for most of the peptides. Again, here with single cell, we had to evaluate how long do we need to inject for, and we can actually inject for 65 milliseconds or maybe 55, somewhere around here. Seems to be the sweet spot for single cells reaching 2,000 proteins. So this was a tenth of what we're running at the moment on our Eclipse. And comparing these two instruments, for single cells with direct DIA, 2,000 proteins on Astral, we get 1,000 proteins on average on Eclipse. And we can boost those numbers a bit with a gas phase or with this 1 nanogram library. But as I showed before, you want to be a bit careful with those approaches. But just to show what kind of numbers you can reach if you're not as interested in the high quality quant. And this one I showed before as well, just to really stress home that with similar runtime, 72 SPD, because we're using an ultimate, it's slower than the Vanquish, so we can do 80 SPD on the Vanquish with the same setup. We really get this good increase of at least 50%, and for lots of cells we're actually seeing a doubling just by running the same cells on the same chromatography, but then combined with an Astral rather than an Orbitrap. So what can we do with this? Can we extract some biology out of this? And one of the applications we decided to look at was cell cycle, because kind of like Ed nicely said, maybe sometimes you shouldn't do single-cell proteomics, and doing single-cell proteomics on a homogeneous cell line doesn't always make a lot of sense, but it was kind of our test system, and we wanted to see, can we extract something out, because cell cycle is happening in these homogeneous cells, meaning there is a level of heterogeneity. And you can see that our data completeness is really quite nice. We have probably 80% or so average data completeness across all the cells. And if we took, so in this case this is 100 HEC293 cells, and we, according to the proteome expression, plotted a PCA, we actually got this circular pattern, which is fully reminiscent of the cell cycle. If we then calculate the spectral angle according to cell cycle proteins, we can actually show that these cells are fully following the standard cell cycle system as we know it. So geminin is a G2S marker, and that's these cells here. PCNA is an S-phase marker, suggesting that S-phase is up here. And if we take the collection of G1 and G2 proteins, you can see that we have the right side and the left side respectively covered by cells that are in those respective states. So we thought it was pretty cool to see for the first time ever cells represented as a circle, reminiscent of the cell cycle. And then we can look at these key components that were not detectable on previous generation mass specs, like the MCM complexes, which is very important for DNA replication and histone recycling. We can see CDK1, which you would expect to be relatively constant in cell cycle, is indeed relatively constant. And we can really start looking at these key distinct subunits of these MCM complexes in different cell cycle states. And this is actually very difficult to do with antibodies, because when antibodies bind, or when these proteins are active, antibodies can't really bind. So you can't really do this with antibody-based approaches very well, but now with Astral we can do it in a non-biased way without any targeting. So I hope I convinced you that Spectronaut is a really versatile DIA software tool that supports all the popular mass analyzers. Not sure what people have in their basements, but indeed all the major ones on the market now are supported. With Spectronaut 17, the Orbitrap Astral delivers double the proteins, double the cells out of the box using our WishDIA approach, and we can now start looking at key cellular decision-making proteins like kinases and transcription factors, and we can even do this at the single cell level now. So we can detect cell heterogeneity at the protein level and dig even deeper into cellular trajectories. And indeed, very exciting room for chemists to become active and bring us new DIA multiplexing capabilities. With that, I would really like to thank the people in my lab, especially Tira, who did the lit DIA work, Valdemars and Pedro, who did all the Astral work, Thermo for the collaboration on the Astral, Biognosis for giving us early access to new releases, Bo's lab, Brian Searle, Ulrich, the DG Proteomics Core, funding, and you for your attention.
Please let's thank together our speakers again. See you in the next time. Thank you.